Yeah, again, thank you, Hongbo, and thank you, Sergey, for inviting me for this conference. I'm not a polymer chemist, but I'm trying, and all the things that I will present to you today is about polymers. In fact, what I want to try to do is I want to try to convince you that we can use small pieces of synthetic polymers. Normally, we just say small pieces of plastic instead of antibodies in similar applications in affinity technology and in biomedicine. Yeah, I just take the laser pointer. Okay. So yes, I will talk about molecularly imprinted polymers, give you some more general introduction and also a few examples on the use of these materials. As you may know, affinity technology is based on molecules that are capable of molecular recognition. I'm sorry, I just need to remove these controls. Okay. Yes. Affinity technology is based on different molecules that are capable of very specific molecular recognition. And the most important class of molecules are, of course, biological antibodies. And because of their property to be able to very specifically recognize other molecules that are called the antigen, they can be used in many applications, for example, in affinity separation or in biosensors, of course, also in immunoanalysis, for imaging, and also in medical treatment. Now, they are biomacromolecules, and sometimes they can have a number of drawbacks if they are used in applications that takes them out of their natural environment, because they tend to be unstable. They are sometimes also difficult to obtain, and all this makes them expensive and difficult to integrate into standard industrial processes. And therefore, researchers have been trying to imagine alternative materials, alternative molecules that can do the same, recognize another molecule, but that may not always have all these drawbacks. Now, I'm a biochemist, so if we look at an antibody from a chemistry point of view, antibodies are proteins, and proteins are actually also polymers. They are polyamides. It's not very nice to say that, and biologists normally don't like when you call a protein a polyamide. Of course, they are very sophisticated polyamides, but let's face it, they have a variety of different monomers, the 20 amino acids, and they are connected with amide bonds to each other. And these monomers are connected actually in a very well-defined sequence, and then the polymer chain folds in space in order to adapt a very specific three-dimensional conformation, which is stabilized by weak bonds, hydrogen bonds, ionic bonds, hydrophobic effect, and others. And in most cases, the conformation is close to a globular shape, with some exceptions, of course. So what could be the answer from a polymer chemist point of view or chemist point of view if we want to do something like that, but without having all these drawbacks that I mentioned for use in technology? Now, first of all, if we want to make a synthetic protein mimic, but we want it to be chemically more stable, we should use a polymer that cannot be hydrolyzed as easily, at least not the polymer backbone. And one class of polymers that you all know that cannot be hydrolyzed is polyacrylates or similar materials that are made by radical polymerization. And in that case, if we say we choose a polyacrylate or polyacrylamide or similar, we have, of course, also a large variety of different monomers with different functional groups. And in the next slide, you will see that we really can mimic the different amino acids that we find in proteins. What is more difficult or almost impossible is to obtain polymerization in a defined sequence. So because radical polymerization is a random mechanism and it's difficult to control the sequence, but what can be done is to arrange the polymer chain in a three-dimensional conformation, which allows also to obtain molecular recognition properties. And this is what we call molecular imprinting, which is a templating process at the scale of a single molecule. So we mold the polymer around single template molecules. Again, if we want that the final material is more stable, we have to use chemical crosslinks rather than weak bonds to stabilize this conformation. And in some cases, if we really want to have a globular conformation, we just have to make very small nanoparticles. So, yeah, just to convince you that we can mimic most of the important amino acids by synthetic monomers. Here is a small selection only. For example, this one acrylic acid has a carboxyl group, is quite similar to, or is the same group as we will find in glutamic acid or aspartic acid, or here we have an imidacyl group like in histidine, or here is this one mimics phenylalanine, etc., etc. So we could probably have an equivalent chemical group on a monomer that mimics any of the natural amino acids. But in addition to that, we also have the possibility of using monomers with chemical groups that are not found in native proteins, but which can be very useful for applications in technology. For example, we can have a more stronger acid, sulfonic acid, rather than just carboxylic acids, or we can have a coupling group for click chemistry. So these monomers, we could call them non-canonical monomers because they have groups that are not present in proteins, but they are very useful for technological applications. So here is a schematic presentation how molecular imprinting works. Before I explain it, I would just like to give some credit to Klaus Mosbach, who is one of the fathers of the technology. There are two main researchers that have worked on the molecular imprinting. One in its beginnings, one is Günter Wolf from Düsseldorf, who is a chemist, so he has more worked on chemical approaches, and Klaus Mosbach, he is a biochemist, and he has basically developed the molecular imprinting that I will present you today, that is based on non-covalent interactions between the target and the polymer. So what do we do? We have this yellow ball here, which is our template. It's the antigen. If we want to use the terms of the antibody world, this would be the antigen or a derivative thereof, which we will mix with monomers. And some of the monomers have chemical groups that allow them to interact with the template. And then we also have monomers that are bivalent that can be used as crosslinkers. So there will be a complex formation through weak bonds, hydrogen bonds, ionic bonds, sometimes also coordination bonds and others, pi-pi stacking, etc. And then we will initiate the radical polymerization, and we will end up with a three-dimensional polymer network that now contains these small cavities. And these cavities are formed because of the presence of the template during polymerization. And they are complementary to the template in terms of size and shape, but also chemical functions. And if we now remove this template molecule, we will end up with an empty binding site that can bind it again very specifically. And this is what we call a molecularly imprinted polymer, or MIP. Before I continue, I would just like to mention that there are, of course, other polymer matrices that we can use for molecular imprinting, not only the polyacrylates or polyacrylamides, but these are about 90% These account for about 90% of the materials that have been described in the literature. Here in the molecular model, this is a little bit closer to the reality. We can see that in the beginning we start with monomers. We have some still flexible polymer chains that are formed. And the more the polymerization and cross-linking progress, we will have a dense network, and the binding sites are actually formed in between the polymer chains in that network. Okay, now in the very beginning, when I started to work on this area in France, we always ask ourselves if we want to use these materials instead of antibodies in a number of different applications that antibodies are used for. The question is, can we really make them as small as antibodies, and can we give them similar properties as antibodies have? And if we look at an antibody molecule, it is about 12 nanometers in diameter. And if we want to be close to that, we have to make really very, very small particles. And the problem with that is that in such a small particle that is only a few nanometers in size, there will be only very few polymer chains. And it is difficult to imagine that you can, in such a small network with few polymer chains and maybe just a few crosslinks, that you can actually imprint a molecular memory in this network, simply because of size limitations. So we thought, okay, let's try it anyhow. And what solution can we find in order to make an imprint in such a few polymer chains? Now, one possibility is to say, okay, if we only have a few polymer chains, what we should do is at least make sure that they are close together, close to each other. So in order to have a higher local density of polymer chains, in order to have a network that can be molecular imprinted. And we made a lot of effort. We synthesized a dendritic polymerization initiator, which is a generation 3 dendron in this case here. And on each of the arms of the dendron, we attached a polymerization initiator. So that's in that way, each of the arms can actually initiate one polymer chain. There are also other versions of this molecule, but here we have, this is the dendron. Here is a central group that can be used for labeling or coupling. And then we have the inverter type polymerization initiator that allow for living, controlled living radical polymerization, photopolymerization. And here at the outside, at the periphery, we can use the groups in order to fine-tune the solubility of the material. So now with these initiators that we call dendrini, we have done solution polymerization under high dilution conditions. And as you can see, the answer is yes, we can make polymer nanoparticles that are of the size of an antibody, because as you see here, the DLS particle size is about 15 nanometers, and this is confirmed by the transmission electron microscope image. So we really get very small nanogels, which are in the size range of an antibody. Antibodies, as I said, 12 nanometers. And also the molecular weight is quite close. Now, normally these nanogels would be not very dense, but because we bring the polymer chains together, we have a density that is close to that of the antibody, 100 kilodalton with respect to 150 for the antibody. So pretty close. And of course, important is that we really can make a molecular imprint. Here the antigen was a small drug molecule called propranolol. Is a drug that is used to treat high blood pressure. And it is a chiral molecule. So we have used the S enantiomer as the template for molecular imprinting. And now if we use a radio-labeled version of this drug at a constant concentration and titrate it with increasing polymer concentration, we really have a nice hyperbolic binding curve and a good difference in binding with the chemically identical control polymer. We call the control polymer NIP, or non-imprinted polymer, and is basically chemically the same material, but without imprints. In the antibody world, this would be called the isotype control or isotypic control. So we have this difference in binding, and we can also check the cross-reaction with the opposite enantiomer, which in this case is 14%. So we also have a good specificity for our imprint. So this was good news, but in fact, we've later found out that we don't need to make so much efforts, as you will see in the other examples that I will show you. We don't need to make so much synthetic effort, and normal nanogel particles of a little bit larger size will work as well. In fact, these polymers, as you know, they're synthetic polymers, and we can obtain them in all kinds of sizes and shapes. The original material that has been used in the beginnings of molecular imprinting by nearly everyone is just a big block of polymer that is mechanically ground to a small particle size of a few micrometer. This is, of course, not very elegant because there are not even two particles that have the same size and shape. But it worked in many applications at that time in the 80s and 90s. And the first paper where Klaus Mosbach showed with some postdocs that they can use molecularly imprinted polymers instead of antibodies in an immunoassay, that was published in the early 90s in Nature. And this was based on this kind of materials. But in the meantime, of course, polymer chemists and material chemists have taken up the area. And therefore, we have now much more elegant methods to synthesize these materials, and we can do all kinds of different shapes. These are our nanospherical nanoparticles made by precipitation polymerization. Here is a porous film with controlled porosity on the surface. These are nanofilaments, nanohairs on the surface. They are about 50 nanometer in diameter, all and a few micron long, all attached to a surface. Here we have small droplets written by a nano fountain pen. They are about 1 micrometer in diameter. But we can also use standard fabrication procedures. For example, wafer scale photolithography, or we can do holographic imaging in films in order to make holograms, or we can use a micro nanoscale 3D printer, as we have here, in order to make all kinds of forms and shapes. So this is the advantage, of course, of using synthetic polymer over a biological macromolecule. But I will give also a few other examples, but the main part of my talk will concern this kind of material, which are small nanogel particles that are quasi-soluble in water. So this is the main material that you will see in the applications that I will show you. Okay, so let's have a look at a few examples, a few application examples, and the first one concerns diagnostics, medical diagnostics. So in that case, we mainly can do two things: either detect soluble biomarkers or do bioimaging and detect the cell surface biomarkers. Yeah, the first example that I would like to show concerns a biomarker for acute kidney injury, and it is a protein called kidney injury molecule 1. This is a protein that normally is found on the surface of kidney cells, but during acute kidney injury, which affects as many as 1 out of 3 patients entering intensive care, and as many as 1 out of 2 patients in reanimation. This is they have an increased risk to contract an acute kidney injury, and this is either related to their illness or to the treatment of the illness, which often affects the kidney. And since these patients will either need a transplantation or need to be or will become dialysis patients, it is of course important to detect and treat acute kidney injury very early. So this is this kidney injury molecule 1, KIM1, is a very early biomarker of acute kidney injury and is found in urine in the case of AKI, of acute kidney injury, whereas normally it is cell surface protein. Now, how can we make molecular imprints of proteins? Proteins are biomacromolecules, and therefore they are not compatible with the standard protocols that people in the molecular imprinting field are used to. And therefore, we can actually again be inspired by nature. If we look at an antibody never recognizes a whole protein. An antibody only recognizes a small part of a protein that is called an epitope. So an epitope can be either a peptide or a sugar on the protein surface, accessible to the outside, and this is recognized by the antibody. So we can also use the same approach, molecular imprinting of an epitope in order to make our synthetic materials. Now, if we have a look at the KIM1 protein, which we have here, we have to choose, of course, the right epitope that we can use for molecular imprinting and that will later be recognized by the synthetic antibody. And for this, we will use rules that are kind of similar, rules or criteria that are similar to the fabrication of peptide antibodies. Now, peptide antibodies, I don't know if you are familiar with the technology. It's based on the immunizing an animal with a peptide rather than a whole protein. And if the peptide is well chosen, it will still produce immunization and formation of antibodies. So our criteria for the choice of an epitope is similar to these peptide antibodies. So we will need accessible epitopes that are protruding from the protein surface and will thus be accessible by the big nanoparticle that we will form. The length of the epitope is between 8 and 20 amino acids. And if possible, there should be no post-translational modifications or disulfide bridges in this area that we will choose. Here we have, for example, these 3 epitopes that look like they are protruding. So we could choose one of them. And of course, we have to make sure that the peptide is absent in other proteins. It must be specific to our protein that we want to recognize. And then once we have chosen an epitope, we need to make sure that the chemically synthesized epitope that we will use for molecular imprinting has a structural similarity to the epitope in the protein. So for example, in the case of this KIM1 protein, if we say, okay, we choose this one, the middle one of the epitopes that we had identified, we will synthesize it chemically, and we have to make sure that it adopts a similar conformation as the peptide in the protein. Now, if the epitope that we chose is a terminal peptide, we can just use the linear peptide for molecular imprinting. But as is the case here, if we have an inner sequence, it's just in the middle of the polypeptide chain of the protein, then of course we have a problem because we need to display the right epitope in the right orientation in order to do molecular imprinting. And therefore, normally we will choose a cyclic peptide in order to present it in the right orientation. So we will take these amino acids and then cyclize them by doing, for example, by forming a disulfide bridge. And in addition to this, in both cases, we have to add a coupling group, and I will explain to you why. Why do we need a coupling group? Because we will synthesize our MIP in a solid phase synthesis approach, which you will see in the next slide. Before we do this, once we have chosen our chemically synthesized, our peptide that we will chemically synthesize and use as a template, we need to do some molecular modeling again in order to verify if the chemically synthesized peptide adopts the same conformation. And there are internet tools, for example, PepFold, that allows to verify what kind of secondary structure your peptide is most likely to adopt. Now, of course, it's only a model. It can be wrong, but at least it gives you some good indication. And in our case here, as we see, we have a large chance that the chemical peptide will adopt a coil structure. So a flexible loop or coil rather than alpha helix or an extended structure. Alpha helix would also be okay, but extended conformation we don't like much because this often means that it is implicated in beta sheet formation. And that is difficult to mimic for the chemically synthesized peptide. But in our case, we have a large percentage of coil, so this is good. And then we synthesize the peptide. Here you have it. And as I said, the synthesis of our MIP will be done on a solid phase. It is by solid phase synthesis, and in order to couple the peptide to the solid phase, we will use click chemistry. In this case, it's alkyne azide click chemistry, but there are many other options. So here we use this strain-promoted version of the click chemistry, but there are, of course, other coupling possibilities. So therefore, we have to add an amino acid that is responsible for the coupling reaction. This is, in this case, a lysine azide, and then the two cysteines in order to make the cyclic peptide, and the rest is our epitope. These are the amino acids of the epitope peptide that we have chosen from the Kim1 3D model. Now we are ready to determine which kind of monomers we want to use in order to recognize these peptides. So we can, for example, use a basic monomer, like this one has an amidine function in order to recognize the carboxyl groups in the peptide. It makes a quite strong salt bridge with also hydrogen bonding at the same time. So this is a strong monomer for carboxyl groups. And then we have, of course, also aromatic amino acids. So we can use an aromatic monomer, phenylacrylamide, and then some more hydrophobic monomers. And the main monomer that we will use is isopropyl acrylamide. And of course, you know what are the properties of polyisopropyl acrylamide or polynipam. These normally can give, if used in the right proportions, thermoresponsive monomers, polymers. Sorry. And this is also important for our application, as you will see. And then we need a crosslinker, methylene bisacrylamide, and the polymerization initiators, which in this case are very, very simple. It's just a redox initiation system with potassium persulfate and TMATs. So all this we need, and now we are ready to do the polymerization. So we take a solid phase, which can, for example, be glass beads, about 100 micrometer in diameter. We activate them, we couple our click chemistry coupling group, and then we click our peptide, which has the azide on the lysine. And now we are ready to directly synthesize around single peptide molecules our synthetic polymer nanoparticles, our molecularly imprinted polymer. Now, since we have a recipe with a large content of NIPAM, we will end up with polynipam, and polynipam really has thermoresponsiveness also in our case here. And the transition temperature, the LCST or lower critical solution temperature, is about 32 degrees. And we will do the polymerization above the LCST, where the polymer is compact, as you see here. So the polymer is compact above the LCST. We do the polymerization, and now before we recover the synthetic antibodies, the MIP, we will first do some washing. And that is the interesting thing for this solid phase synthesis, like with all solid phase synthesis, by the way. We can wash out all the unwanted parts that we don't need, unpolymerized monomers and particles without imprint, etc., etc. So we can really do extensive washing at 38 degrees, and then we just have to lower the temperature below the LCST, for example, to 4 degrees, and the particles will swell in come off the column, and we can reuse the column. This is the best also. If the peptide was expensive, we can reuse it. And this gives us molecularly imprinted polymer or MIP nanogels that we can use as synthetic antibodies. So we always do some characterization in the beginning. We check the particle size, which is about 50 nanometers. We do also control polymers. We call it control MIP because we cannot make really a non-imprinted polymer. We cannot make on the solid phase synthesis a non-imprinted control polymer, but we can make an imprint against another molecule, a smaller molecule or another peptide, which we then use as a control. And then the second verification is that the particles can bind the template peptide. This is the first sign of success. If molecular imprinting has worked, this is if the polymer binds the template peptide. So we just click a fluorophore, for example, fluorescein or rhodamine or some kind of fluorescent dye to our peptide, again with click chemistry, and we do a similar binding assay as I have shown before for the propranolol. So we take a constant concentration of the fluorescent peptide and incubate it with increasing concentration of antibody. And then it will, if everything works, we will have a hyperbolic binding curve, and the control polymer should not bind if possible, which is the case here. So imprinted against a different molecule, there is no binding. Now, of course, we need to verify as well if we really have a specificity, if the polymer is specific for our target, and this can be easily done using a competitive binding assay. So what we will do is we will keep the concentration. We will again use the fluorescently labeled peptide, and we will keep its concentration constant. For example, I don't know, in a low nanomolar concentration, 4 nanomolar, something like that. And then we will also add a certain amount of molecularly imprinted polymer, which will also be constant. And then we add, in addition, non-labeled peptides or the whole protein or other peptides in order to see if they can displace the labeled peptide from the binding site in the MIP. And we look at which concentration we get about 50% of displacement. It's called the inhibitor concentration of 50%. And the lower this number is, the better. And of course, we will first of all verify if our template peptide, which is not labeled, can displace the labeled template peptide, and we put this binding to 100%. And now we can do the most important test. The question is, does our synthetic antibody also recognize the whole protein? Otherwise, the whole work will be useless if we don't recognize the epitope in the whole protein. And as you can see here, the IC50 of the KIM1 protein is very similar to the one of the peptide. We have a cross-reactivity of 92%, which shows that we recognize indeed the whole protein, and we can use our MIP as synthetic antibody. We also check the linear epitope, which can fold into the binding site, and it will also bind. But if we use a different peptide, a circular peptide with similar amino acids, there is no binding at all. Now, more recently, we have had a collaboration with NMR chemist from Sanofi in Germany, in Höchst, and he proposed to use saturation transfer difference NMR to obtain an additional proof of the existence of molecularly imprinted binding sites in these materials, because we know the particles bind our peptide, but we don't really know if we have binding sites. So how does it work, this STD NMR? We will mix our polymer particles together with the peptide, and then we will irradiate the polymer with a radio frequency pulse train, and at the frequency that is only absorbed by the polymer, but not by the peptide. So this is something where we have to choose very carefully. which frequency we can use in order to really only be absorbed by the polymer. And what will happen is, if the polymer binds the peptide, it will transfer some saturation of the electromagnetic energy into the protons of the peptide. And if that happens, that means the peptide really is bound tightly to the polymer particles. Otherwise, this saturation transfer to the peptide protons would not be possible. So, yeah, this is done first at the frequency where we have a resonance, so where the protein absorbs, and then we do it again at the frequency where nothing absorbs. And we have to avoid any frequency where the peptide absorbs, of course, because we only want to see the peptide saturation by the transfer from the polymer. And then we do the different spectrum, and this will be our STD NMR spectrum. So as you can see here, we have the template peptide alone. This is just the STD spectrum of the template. And since we use the frequency where the peptide doesn't absorb, we will not have much signal. But now if we add a little bit of MIP, the saturation of the MIP will be transferred to the peptide, and suddenly we see the peaks of the peptide appearing little by little, because this is a cumulative effect. The longer we wait, the higher these peaks will become. Now, if we look, if we take a look at the control, now we use the control MIP, which is not specific for this peptide. We don't have this effect. The peaks stay small. And if, as I said, the linear peptide also binds, so it also works in the same way with the linear epitope. And now if we use the non-template peptide, the control peptide that I showed, nothing happens again. So we don't have any STD effect. So this really shows this, and here this shows that we have a very tight binding of our peptide in a cavity in the molecularly imprinted polymer synthetic antibody. And in fact, this technique has a atomic resolution. We can even through the spectrum identify which of the amino acids are involved in the binding that are very close to the synthetic antibody to the polymer network. I mean, these peaks, for example, are of protons coming from tryptophan and phenylalanine. And the STD NMR is a little bit more sensitive to hydrophobic amino acids, and there is a complementary technique called water loxy where we can see more easily the hydrophilic ones, and we have used that as well, but I will not detail it here. So this is actually was very good news, because it really allows to verify the existence of specific binding sites in these small particles. Now, let me see what we can do with these particles. Can we use them to detect the KIM1 protein biomarker, for example, in biological samples, in urine or elsewhere? And for this, we have the possibility, since we have a synthetic polymer, we can do a lot of engineering. And one thing that we can do, we can incorporate a reporter group. So a reporter group, for example, a fluorophore, which when it interacts with our target protein, with the KIM1, will change its fluorescence. So here's an example of such a reporter monomer that can be copolymerized in these nanogel particles. It has a polymerization part here, and then it has the fluorescent moiety, which is a naphthalimide fluorophore. And here is the piperazine. This is a recognition group that allows it to interact with our peptide, in particular with carboxyl groups in the peptide. And this fluorophore has a particularity. This assembly of fluorophore and recognition group, it has the particularity that when there is no interaction with another molecule, the two electrons here, they will be transferred to the fluorophore and will quench the fluorescence. It's called photoinduced electron transfer. Because of this electron pair, the fluorophore actually has very little fluorescence. But now if we add our target molecule, the epitope peptide, which has carboxyl groups, this electron pair will be occupied and the fluorescent quenching will be removed and the monomer becomes fluorescent. And as you can see here, we really have, with increasing concentration of peptide, we have increasing concentration, increasing intensity of the fluorescence signal. Now starting at the below nanomolar concentrations up to here is up to 30 nanomolar. So this graph here could directly be used as a calibration curve in order to measure simply by adding these molecularly imprinted, let's say, nanosensors into a sample, from a patient and measure the fluorescence compared to a standard. So this is very nice, and it shows that there is really a potential to use these materials in bioanalysis. Now I want to make just a very short parenthesis, because we are talking a little bit about sensing. I will, for a very short moment, leave the nanogels and look at a few other materials with you in order to show that we can really make molecularly imprinted polymer-based sensors. And there is, I have only two examples. The first one is based on an optical fiber, a standard telecom optical fiber with 8 micrometer core. And we, at the end of this fiber, the fiber is cleaved, and then at the end of the fiber, we will synthesize a molecularly imprinted polymer tip. And how can we do this? We just have to dip the fiber in the monomers and then shine a laser light into the couple a laser into the fiber, and when the laser, where the laser comes out, it will start the photopolymerization of our molecularly imprinted polymer in form of a tip. And if we use a bifurcated fiber, we can actually even use the other arm of the fiber in order to do spectroscopic measurements through the fiber. So here you can see, this is the cleaved fiber. This is the drop. We just need to put a little drop of precursors, polymer precursors, and then we start the laser, and we will end up with a very small polymer tip, which you have here. This is a confocal microscope image. It only has fluorescence outside because we actually do a core shell format. So we make first a tip, which is normal polyacrylate. This is the optical element, and then we dip this tip again in the mixture of monomers, and this time we use the monomers that we need for molecular imprinting. So we get a very thin layer of molecularly imprinted polymer around this micrometer-sized tip. So how can we use it for measurements? We can, for example, again, introduce this kind of reporter monomers, and if we do this, we can directly measure analytes, even if they are not fluorescent themselves. So in our molecular imprinted polymer mixture, we have the responsive reporter monomer, the naphthalimide monomer, and here we have our target molecule is a small molecule, is a herbicide, 2,4-D, 2,4-dichlorophenoxyacetic acid, and it has a carboxyl group. So if it interacts with the dye in the polymer tip, the fluorescence will be enhanced, and that is again what we can see here. We have even at very low concentrations, at 0.25 nanomolar, we have a fluorescent enhancement in the presence of the 2,4-D. The limit of detection is 0.25 nanometers if we add some gold nanoparticles to the mixture. So this enhances a little bit the signal. It's not a plasmonic enhancement, they just work like nano mirrors, and more light is reflected back into the fiber. So we have a limit of detection of 0.25 nanomolar for the 2,4-D, and a good discrimination of similar molecules that have a structure close to our target. So this was one chemical sensing example that I wanted to show, and I continue with a second one, which has been made by a different approach. This time we use again photopolymerization, but in order to write directly, like in a 3D printer, very small polymer features. We have used this technique that is called two-photon stereolithography. And yeah, two-photon stereolithography is a very specific technique that allows to write true 3D structures. It is based on the fact that normally, if we use a photo initiator, it will absorb one photon and then it will be set off and start polymerization. But if we would do this for photolithography, we would polymerize everything in the solution that would be in contact with the laser beam. And if we use two-photon polymerization, then the photo initiator needs to absorb two photons in order to be set off. So we can use light with less energy at higher wavelengths, for example, near infrared light. And if the photo initiator absorbs two photons of this wavelengths, then we will have enough energy to set off the polymerization. And in order for this to be possible, we need two things. Our laser impulse needs to be very much focused in time, but also in place. So we need to have a pulsed laser, femtosecond laser, in order to have a very short pulse with very high intensity, and then we need to focus, of course, the laser beam in the solution. Now, for example, we can put a droplet of the monomer solution and just focus our laser beam in a very small spot, which is called the voxel, which is about 200 times 600 nanometers in size. 200 times 200 times 600. It's a little bit oval shape, like an egg. So if we do this, then only in the focal point of the laser and only when we have a pulse, now we have a high enough intensity in order to go over a threshold and be able to have two photons absorbed by the initiator instead of the polymerization. And if we do this, then we can, of course, avoid polymerization all the rest of the solution, and we can write very small features. Here we have these photonic crystals at a very good resolution, or we can also, this is a 3D structure, but we can, of course, also write the 2D structures on the surface. And here we have actually written small cantilevers that are used as chemical sensors. So we just write a socket, and then these cantilevers are made out of MIP. And they are put into oscillation by the piezoelectric effect. And now if our analyte molecule, which is again 2,4-D, adsorbs to the cantilever, it becomes a little bit more heavy, and the oscillation resonance frequency will change. And this is what we can measure. So as you can see here, in the presence of 2,4-D, we have a very large drop in oscillation frequency of our cantilevers. So these are small sensing elements, just a few micrometer in size and very thin, just a couple of micrometers thick, and they can be used as chemical sensors. Okay, so this was just in order to show you that we really have a very versatile materials, and the engineering of these materials is very easy. But let me come back to our nanogels that we want to use in chemical diagnostic. I also would like to show you that we can do bioimaging with these materials. And for this, in the beginning, we have used cell surface glycosides as molecular targets, for example, sialic acid or hyaluronic acid. Now, these are on the surface of many animal cells, and if the content of these surface glycans changes very much, this could be an indication of a transformation of the cell into a tumor cell. So we use these as targets. For example, the hyaluronic acid, we just use a monomer of it, which is glucuronic acid and N-acetylglucosamine. So we made an imprint against glucuronic acid in order to use, again, the epitope imprinting approach and make synthetic antibodies. So yeah, template glucuronic acid, we use it with a coupling group that allows it to immobilize it on our solid phase, because we will again use solid phase synthesis for synthesizing the MIP. Here is the binding assay that I showed already before. We compared to the non-imprinted polymer, and we check the diameter of the particles, which are about 60 nanometers in this case. And now we are ready. The only thing that we have to do, we have to add to our monomer mixture a fluorescent monomer in order to have fluorescent particles, and they can directly be used for bioimaging. So the protocol would be the same as for antibody bioimaging. We fix the cells. It also actually works with live cells. We plot non-specific sites. We do maybe some enzymatic treatment, and then we incubate with our synthetic antibody and look under the fluorescent microscope. And here is the image that we get. So as you can see, we have a very good resolution of the surface of the cells. Because of the small particle size, we cannot see individual particles. We have a very nice staining of the glycans on the cell surface. And if we compare with a natural molecule that has affinity for hyaluronan, the hyaluronan binding protein, we have exactly the same type of staining. The blue is the nucleus staining in order to know where the cells are. If we do exactly the same with the non-imprinted polymer, which is chemically identical, we have still the nucleus staining, but we don't have the cell staining, which is normal because there are no imprints for hyaluronic acid. Okay, and of course, what is interesting in this context of bioimaging is, of course, not only cell imaging, but in particular tissue imaging. And here you can see that we can use these synthetic antibodies for bioimaging of skin tissue, for example, here that we have obtained from a hospital. Now, as I said, these polymers are synthetic polymers, so engineering is much easier than with biomacromolecules, with real antibodies. And here is just one example. Also in the context of bioimaging, you probably know that fluorescent imaging is often done now with fluorophores based on fluorescent nanocrystals like quantum dots or upconverting nanoparticles. And some time ago we thought, okay, why can we not use the light emitted from single quantum dots in order to synthesize a very thin layer of molecularly imprinted polymer directly on the particle surface? If we irradiate a quantum dot, for example, with UV light, it will be excited and it will emit visible light fluorescence. This one is an indium phosphate zinc selenide quantum dot, and they, depending on the size, they emit, for example, at 540 nanometers. So what if we use this light, the fluorescent light, from the particle in order to polymerize by photopolymerization a thin layer of molecularly imprinted polymer? So for that, what we have to do is we have, of course, to find a photo initiation system, which is set off by this wavelength. And there is this type 2 photo initiators that are used together with an amine that are based on a dye. Here it is eosine, and this eosine absorbs exactly the wavelength that is emitted by the quantum dot. So this is very nice. So this would mean that if we add our monomers and this dye plus an amine, we shine light on the quantum dot, we will have photopolymerization of a polymer layer around it. And this actually really happens. For the moment, we don't use monomers for molecular imprinting. We just make a thin polymer layer to stabilize the quantum dots. Now you can see here a little bit the contrast between the polymer outside and the quantum dot inside. And now we can put all the other monomers in order to do molecular imprinting, so glucuronic acid and cross-linker and functional monomers, and switch on the light again. And with some more fluorescent light of the quantum dots, we will make a second layer, and this layer now is molecularly imprinted polymer. So we end up with a nanocomposite material with molecular imprints at the surface of a diameter of about 100 nanometer. And again, can be used for bioimaging. So we actually have mixed two different sizes of quantum dots, one emitting in the red and one emitting in the green. And around these we have synthesized MIPS for two different molecules, glucuronic acid and sialic acid, and you can see we can use them together because the excitation wavelength is the same. We can use them together, and there are some parts of the cells which are more green and some are more red, depending on which polymer, which glycan is situated. Okay, so now this was this was an example on diagnostics, and I want to finish with some mention of treatment. We can also use them as antibody therapy, these materials, or at least we think we can do this. And in order for this to check, we have used a target molecule which is called caterins, cell surface molecules that are responsible for the interaction between cells. And these are calcium-dependent adhesion proteins, and there is some epitopes that are responsible for the interaction between the caterin of one cell and the caterin of another cell, and that's how the cells form these adherence junctions. So what if we imprint these epitopes? Then we can maybe block the interaction between cells, and this is interesting in therapy, because there are actually antibody therapies against the caterins in order to improve chemotherapy of certain cancers. So here is, we chose one of these peptides, the linear one, the other one would be a cyclic one. We also did with the other one, works the same, and did again the same approach. So choose the monomers, do solid phase synthesis of our imprinted polymer nanogel particles. Particle size is again around 50 nanometers. Binding is good. Difference with the non-imprinted control. And here we can see again what is important. The antibody needs to bind not only the template peptide but also the whole caterin protein, which is the case. And here you can see a little bit the specificity that we can reach. If we change just one amino acid in the peptide, there is no binding. This tryptophan, or here the proline, if we change a proline against alanine, there is no binding at all. So this shows that the imprints are really very specific. And we can again do bioimaging, which I have already shown. It works very nicely with the imprinted polymer. And if we remove all the proteins from the cell surface, we have no imaging at all. But the question was, can we inhibit cell-cell interactions? And there is a test for it. It's called cell aggregation inhibition assay. If you take cells and you seed them in cell growth medium, because of the caterin interactions, they will form dense carpets, and this we can completely inhibit with the addition of molecularly imprinted polymer. But if we block the molecularly imprinted polymer by adding the free peptide, the polymer doesn't work anymore. The binding sites are blocked, and that's why we still have we again have the dense cell carpet. And what if the cells already have interacted? Can we loosen the interaction between the cells once they are formed? And here you have an example. We have used spheroids. Spheroids are small aggregates of cells, mostly used with tumor cells, but also with other cells. And they are considered a very simple model of tumor tissue. So we use tumor cells, HeLa cells, to make these spheroids. And as you can see, if we add even small concentrations of molecularly imprinted polymer, we will completely loosen up the interaction between the cells, and we will loosen up the tumor, which is good for combination with chemotherapy, because the agent will be able to reach the inside of these tumors as well. And I don't want to detail much this part, but caterins are also responsible for invasiveness of tumor cells. And if the question was, can we block the invasiveness with our synthetic antibody? And there is also a test. It's called the Matrigel invasiveness assay. And as you can see here, if we add to this test our molecularly imprinted polymer, the invasiveness index of the cells is much reduced. Okay, last but not least, I want to show you also an example of everyday's life. And a few years ago, the L'Oreal, the cosmetic company, asked us if we can help them to make a new type of deodorant principle. And in fact, your body odor appears because we have these non-odorous precursors. This is an amino acid, asparagine, and then it is conjugated to odorous short-chain fatty acids. And bacteria on our skin, these molecules are secreted by our skin through the sweat, and the bacteria, they eat it and they liberate these odorous acids, and this is how the smell, the body odor appears. So the idea was to sequester the precursor. Before the odor appears, to sequester the precursor and see if we can in that way inhibit the formation of the odorous acids. So we used a template, which is an analog of the molecule, and made the molecular imprinting of this complex. And the idea is then to prevent, to sequester the precursor and to prevent the bacteria to hydrolyze the molecule. And as you can see here, it works very well. Always much better with the imprinted polymer than with the control polymer. And really, there is complete suppression of any body odor if these materials are used. And funny enough, this was probably the occasion where we had the most general public attention of all our projects, because this is something that people really need. And I think this is also one of the take-home messages, that we need to do something that is needed by the people, not only things that are nice scientifically, but also something that has a lot of general importance. So yeah, to summarize, molecularly imprinted polymers are antibody mimics. They are tailor-made and they are more stable. Otherwise, have similar properties to biological antibodies, and can be cheaper and more easy to engineer, and can be used in many different applications, for example, in diagnostics and biomedical therapy, but also many others. And I would like to thank all the people from my group that have over the years contributed to this, and also all the funding agencies and companies. And I thank you for your attention.